Welcome everyone. Today I going to discuss about human cloning, genetic unit and biotechnology, DNA gene cloning and pharmaceutical product. This is some of the discovered as a defense mechanism in the bacteria used to cut visible element of the DNA work into two ways exclusive, inducive, both type recognize unique specific sequence. This has induces cut DNA at a specific sequence. Gattaca restrictions. These are the basic info. Organization of the piece of DNA that will carry instead DNA from another organism should be a small circular piece of DNA that will reproduce quickly and carry a selective advantage. Bacterial, plasmid, and nucleus virus and the instead are also used. Plasma are used to replicate and recommendations. Procedure use same restrictions, mix gene with the vector in the test tubes, add DNA lasers to beam gene and vector, place plasmid and bacteria, place recombine molecules in bacteria, grow bacteria on the path with the antibodies, only bacteria with plasma will grow. Cloning gene, brocotypes, urotypes. Reverse transcript, once cDNA is produced, grow clone bacteria. Isolate human cells and grow tissue culture. These are the some very basic example and information we are observing. Biotechnology, defined broadly, is the engineering of organism for useful purpose. Often, biotechnology involve the bacteria of hydrogen and their introductions into organization in which some or all the gene is not normally presented. What is cloning? Any manipulations of an organization genetic material to produce a new organization or organic produce included in the definitions are genetic engineering, clone biotechnology, and biomedical. Element cloning. Why animal cloning? This is some basic example of animal cloning. The biotechnology of reproductive cloning, even under the best of circumstances, the curricular technology of the cloning is very inefficient. Cloning provides the most direct demonstrations that all cells of an individual share a common genetic blueprint. May be completed in a party dishes needed are eggs and mature skin cells. Each eggs contain a nucleus inside of the zona in an immune eggs called a polar contain a nuclear how to carry out the process and all the left in the zona. Fabric in the injections in the newly fused eggs are then given an electronic show the skin cells in, cellular is now in cytoplasm. The nuclear transfer process, the benefits, and many interesting aspects we can figure out. This is some of the very basic improvement of the genetic body. Some thorny ethical question: are the masses of the cells is human? Is it ethical to harvest embodied cells from the extra created during the virtual cells from any sources? Then you are on integrated twice present problem of rejections. Based on the fact that each individual has his or her own unique DNA, functional genes, intros are unique intro content sequence, both lengths of sequence and him to reproduce terms the variation number are not even the same in the identical twins. So this is the very basic questions about the genetics engineering, and if we can follow this genetic engineering process, we can grow GMO-free Europe. So this is the basic southern and northern involved in the following procedure: cut nucleic acid with the restriction enzymes, dinner to make single strands, run on gels, blot nucleic acid from the gel onto the filter paper, use radioactive problem to look for the gene of the choice. Not blotting will allow you to find a genes matching mRNA sequence, can then make cDNA from the mRNA. These are the very basic informations and we have to understand and we can take necessary initiative. If you have any question, you directly can communicate with me. Thank you. Bye bye and take care.